Hey guys, I'm Diola. I'm a part of the Fenton Lab at the Food Science and Human Nutrition Department at Michigan State University. This is basically my experiential learning presentation. So I'm presenting to my lab group mates, Travis, William, and Celine, and then to you, Dr. Becker, behind the camera. So yeah, this is my presentation. It's titled Polyphenolic Profiling of Grass-Fed Beef and its relationship to beta-carotene. So I guess we'll get started. So here's my table of contents. We're first going to start talking about polyphenols, their structure and their function. I'll give you a little background. And then we're going to dive into what I did as a project, give you the hypothesis, objectives, give my experimental design, and then eventually get to the results, conclusion, discussion, and then future directions. Okay, so what are polyphenols? I guess polyphenols are these huge ring structures. They have a lot of aromatic groups, which are basically huge carbon rings, these cyclic molecules basically. And there's hundreds of classes of polyphenols. They're basically secondary plant metabolites within the plant kingdom, and they're thought to have these anti-inflammatory, antioxidant effects. Basically, they protect plants from stress in the outside world, and there's a lot of studies that are shown to that prove that polyphenols can inhibit inflammation or oxidation in vitro, and there's also studies in vivo of polyphenols interacting with inflammation as well. And then you can find different classes of polyphenols in different groups of foods and plants, so that's a little background. And then we'll dive further, so we're going to start talking about specific classes of polyphenols that I looked at in my research. So the first polyphenol we're going to talk about is quercetin. They're under the polyphenolic class flavonoids. There's a lot of research studies on it that show it has anti-inflammatory, anti-carcinogenic effects and how they interact with the immune system. A lot of them are found in vegetables and fruits. Some examples are red wine, onions, green tea, etc. Again, there's a lot of in vitro studies on how quercetin actually binds to neutrophils and macrophages and inhibit them from producing some cytokines, pro-inflammatory cytokines. There's limited studies of quercetin inhibiting inflammation in vivo, but there's more interest in that. And then I gave some examples of like serum TNF-alpha, which is another pro-inflammatory cytokine. IL-1 beta and IL-6 were reduced by quercetin in rats, so that was another study I looked at, and here are just pictures where you can get quercetin. So this is the first polyphenol I looked at. Second one's gallic acid. So gallic acid is found in a lot of herbs and foods. They're called hydrolyzable tannins. You can find them in blueberries, walnuts, flax seeds, etc. And gallic acid has a little bit different effects than quercetin. They kind of have an anti-hyperlipidemic effect. Basically, if you consume these, you might see a decrease in LDLs and triglycerides and an increase in HDLs. So there's a lot of studies with gallic acids and its intervention with cardiovascular disease. So that's gallic acid. And then we're moving on to proanthocyanidins, or tannins. These are specific, another specific class found in plants. You can find them in cinnamon, grapeseed extract, green tea, black tea. So if you go on like any like health.com or whatever, you're going to see that tannins are in a lot of teas. So that's why they're saying it's healthy to drink tea because of tannins. They're known to enhance aroma and mouthfeel, and they're basically called oligomeric flavonoids. So what I was talking about, quercetin is a flavonoid, but tannins are basically big oligomers of flavonoids just bunched together. So like a lot of quercetin, a lot of gallic acid just hydrolyzed together. They consist of catechins, condensed flavonoids, like what I said, gallic acid esters, and they're just huge ring structures, and they're basically polymeric building blocks. And more research is being done to see its effect on, again, cardiovascular disease and urinary tract disease. And then finally, anthocyanins. They're actually acylated forms, sugar forms of anthocyanidins. So anthocyanidins are basically anthocyanins without the sugar. They're found in a lot of brightly colored fruits and flowers, so your grapes, there's a lot of gene expression with that. They're actually expressed in autumn leaves, and they're not a carotenoid, but they're actually expressed before the autumn season in the summer, preparing for the fall and the colder weather. So there could be a role of protecting the leaves in the fall and the colder weather. Yeah, like what it said, it's produced at the end of the summer, and again, it kind of has the same properties, anti-inflammation, etc. So we're going to get started talking about my project. So we were interested in looking at beta-carotene, so I'll give you a little background on that as well. It's pro-vitamin A, potent antioxidant. It has a red-orange pigment color, and again, it's found in a lot of fruits and vegetables, especially known in carrots or the GMO gold rice, where yeah, the rice was gold, and they changed its genetics for beta-carotene. So basically, I'm going to start diving into my research. We want to see if we can find polyphenols in beef tissue. So our hypothesis is the supplementation of complex forages within the diet of beef cattle. We like to think that we can find these phenol compounds that I was talking about earlier within the beef tissue. And since I gave examples that phenols are in a lot of plants, we think that supplementing these cattle with different kinds of forages can lead to higher amounts of polyphenols within the beef. So basically, our lab has studies on these types of beef that were fed with different kinds of forages, and the beef were profiled for different macronutrients and micronutrients, and beta-carotene and omega-3 fatty acids were two of those nutrients that were profiled. And since beta-carotene and omega-3 fatty acids have similar properties to polyphenols, similar structure and properties, we believe that higher amounts of beta-carotene and omega-3s result in higher amounts of polyphenols. So I'm gonna go to the next slide. So here's the chart of the different kinds of feeds we gave to the cattle. This is actually from Lake City, Michigan. It's one of the MSU farms, and basically these are different forages that were fed to the cattle, and that's what the research is about. We might find differences in phenol content. And basically, we have these beef samples already, and we already profiled them for beta-carotene and omega-3 fatty acids. So we're going to separate them into two groups, N equals 10 for a high beta-carotene and omega-3 fatty acid group, and N equals 10 low beta-carotene and omega-3. And then from there, we're going to develop a method for extraction and separation of polyphenols from the beef tissue, and then evaluate between the two high and low beta-carotene groups. So here's the other table to see how the significant differences between the two groups are chose. So extraction, I'm going to start diving into the methods. It just consists of a mechanical breakdown of the beef and then adding a methanol to get these phenols into solution if there's any, and to really break down all the beef and the macronutrients and micronutrients. And then from that extraction, we go straight to this thing called reverse phase column chromatography. So essentially, chromatography is you have this column, and then there's two phases. There's a mobile phase and a stationary phase. And what we do in our lab is called the C18 silica stationary phase, and it basically collects nonpolar and partially polar molecules. So polyphenols are really nonpolar with those huge carbon rings, but they also have some polar properties with some oxygen and hydrogen. So when we put our beef extract onto the column, they should adhere to those beads in the column and basically capture those polyphenols. And then from there, the different characteristics of the classes of phenols I showed should be pulled off from the stationary phase with a strong mobile phase. And we're going to talk about that more. And I like to point out that pH is extremely important within column chromatography. because a change in pH can really mess up if the molecules adhere to the stationary phase or not. So here are the different mobile phases. So like what I said, the gallic acid, anthocyanins, quercetin, peroxycyanidians, they have different mobile phases to pull out those phenol classes within the column. And so yeah, so you can look there, quercetin, ethyl acetate. So that's really to pull out those different classes from the column. So then from there, we're going to go to UV-vis spectroscopy analysis. So there has to be further treatments done to get it prepared to go on to the UV-vis. We're going to use a 96 well plate to basically analyze our samples, and then we're going to plot that against a standard curve to get milligrams of phenol, of the phenol classes per mil, and then we're going to extrapolate that back to get it to milligrams per gram of beef, which is basically serving size when you go to 100 grams. And then, yeah, so those are the different treatments to get it ready for analysis. So we use Folin for gallic acid. There's aluminum flavonoid assay for quercetin, which invokes a yellow color. Acid catalyzed butanolysis is for proanthocyanidins. And then the pH differential method for anthocyanins, which is still being optimized right now. And then here are the different absorbances that the phenol classes basically absorb at. So you can just look at that. And then for statistics, all statistics so far have been conducted in PRISM. We use the Wilcoxon test for non-parametric sample sizes. And so here's some of the results that we got. These are still preliminary results because we haven't really finalized. There was solid data, but a one-way ANOVA was conducted on this between the two groups and a positive control in cherry. So you can see the significance here for gallic acid, and then this was the quercetin. And yeah, you see a significance between the cherry and the two groups. And then we went.
I have a question.
Yeah.
Previous slide. So there was no significant difference between the quercetin equivalents between the high beta-carotene and cherry.
Mm-hmm.
There was no significant difference.
The high beta-carotene and cherry.
And cherry.
Yeah. So I guess within our research so far, at this point we didn't optimize the treatments post fractionation, the column chromatography. So some of the data is kind of skewed, I guess.
Okay.
So right now we're still developing the methodology for that. So that's why we weren't able to see a difference, and that could be one of the reasons why.
I have a question too. Why did you choose cherry?
So cherry is we actually researched like different manuscripts, and we're able to see that quercetin in cherry is detectable.
Okay.
There's actually a lot of phenols, like what we were talking about, how there's a lot of phenols in plants and fruits. So cherry is supposed to have it. So we use that as a positive control since other papers already did that.
Okay.
But some of this data could be skewed just because we didn't really finish the methods yet. But if we take that a second step further, we actually looked at season. So for season plants, we like to believe that in the springtime, since the sun's out, more nutrients are in the plants instead of being held in like in the fall. So we think that in the spring, cattle that are finished in the spring with these different forages, that there might be higher phenol content. So we could see that in the quercetin graph, that there was a statistically significant difference when comparing the seasons, fall and spring. So that could be another angle to look at when feeding your cattle. Yeah, so conclusion, discussion, we basically, from those results, those preliminary results, we saw that phenols can be detected in beef tissue. The significance is that this is the first method developed to extract phenols from beef tissue. We basically need more experiments before we can make this into publication. And we already finished the quercetin and gallic acid assay, and the other classes such as anthocyanins and prothocyanins are still being developed. Here's some pictures of smiling beef. And future directions, again, more experiments needed, and we need to finish the methods for reproducibility. And here's a picture of A5 Wagyu Japanese beef. There's a lot of marbling, it's pretty beautiful. So. Does anyone have questions? Will?
What was your favorite part of this project?
My favorite part of this project is really just the methods, I guess. It's really cool because this is, especially for the UV part, you really get to see the color of the different fractions. So you can really tell when some sample is higher in phenols or not, because they have higher yellow color or higher blue color. And it's just really cool handling the samples and having that feeling of, oh, like there are phenols in here, and it's really groundbreaking to be able to have that feeling, I guess.
One question. Do you think that if they find high phenol content in beef, it'll take away some of the stigma of beef being bad for your health?
Yeah, so there's a lot of nutritional, obviously, immunology that we talked about. And yeah, I mean, this could be the first step of destigmatizing red meat. I think red meat, it's hard to have studies and have a control for that, since you can't have someone eat red meat by itself for the rest of their life. So far with the studies I've seen, there's only correlation, not causation. So I think this study could give you a different angle, that if we are seeing these anti-inflammatory molecules within beef, and we can treat our cattle or feed our cattle in a different way to increase these molecules, it could give someone a different outlook on nutrition, instead of just going, just completely canceling red meat overall. So someone who still wants to eat a steak, could look at eating grass-fed beef or something. So yeah.
Sounds cool.
Anyone else have questions? And then here are my references. Thanks, guys.
Good job. Nice job.